Hello bio students, this is Praful Sharma. Welcomes you all in this session where we are going to sum up the chapter Enzyme. In this chapter we discussed many subheadings like introduction of enzymes, their structure, properties, mechanism of enzyme action, their nomenclature, mode of enzyme action, and finally the factors affecting enzyme activity. So let's start with the introduction. Enzymes, as we know, they are completely proteinaceous in nature and they are biological catalysts also, means they behaves as a catalyst. They affects the rate of reaction and the reactions are biochemical reactions, means they used to take place within a living body. Structurally, enzymes are tertiary or quaternary form of protein and have active site. This active site is actually further divided into binding site and catalytic site. Binding site is the site where the substrate will bind itself, and once it gets attached with the binding site, the catalytic site starts catalysis. Enzymes are completely protein, but sometimes a bit part of them is designed by some non-proteinaceous things also. That non-proteinaceous substance is a cofactor. So a cofactor can be organic or inorganic in nature, and this is how an enzyme gets a complete structure. Zinc, magnesium, etc. are inorganic substances and they may be used as a cofactor by some proteinaceous enzyme to activate themselves and perform their catalytic activity. In that case, it is inorganic cofactor. Organic cofactors are organic substances which might be attached with the proteinaceous part of an enzyme so tightly that it's not easy to get them detached from the original structure. Then in that case we call them as a prosthetic group. On the other hand, if the organic substance attaches very weakly with the proteinaceous part of an enzyme, that it is easily reversible or we can detach it from the proteinaceous part very simply, in that case it is termed as coenzyme. So when a protein is without its cofactor, we call it as an apoenzyme, and the cofactor we already named as cofactor, whether it is organic, inorganic, whether it is prosthetic or coenzyme, depends. But the proteinaceous part is named as apoenzyme, and when apoenzyme attaches with the cofactor, the whole structure is called holoenzyme. In biochemical reactions, the reactant is termed as substrate, and when a substrate binds to an enzyme, it forms an enzyme substrate complex. This complex is reversible, that means the substrate may get detached from the. And now we know that enzymes are proteinaceous in nature and proteins are synthesized by ribosomes. Ribosomes are found within the cytosol as well as they are attached with the surface of endoplasmic reticulum. So this is the site where generally the proteins are designed, and once they get modified into their tertiary or quaternary forms, they are termed as enzymes, which are quite active in a biochemical reaction. Naming an enzyme was quite very simple in earlier time, when people used to name an enzyme according to the substrate it catalyzes. Some enzymes were named before a systematic way of naming enzymes was formed. Say for example, by adding a suffix ase at the end of the name of a substrate, enzymes are named. Such for example, like if an enzyme works on maltose, then the name of that enzyme was named on the basis of this substrate, and the suffix ase is added to the substrate name and it became maltase. So maltase is the enzyme which works on maltose. In this way, earlier the name was designated. So we have a number of enzymes like lactase, maltase, cellulase, lipase, amylase, protease. All they are suffixed with ase, and the name was based on the substrate they are working on. Enzymes are specific for some properties too. Say for example, protein nature. Enzymes are chemically protein that we already know now. They have colloidal nature also. That means because they are too big in size, due to their large surface, they provide extensive surface for biochemical processes. They are specific for their reaction. Usually one enzyme catalyzes only one reaction for our functional or new specific chemical bond. Enzymes have major part of an enzyme is made up of protein, and protein being amphoteric in nature, they can react with both acids and basic substrates. As we already know that the enzymes are too big in size. Say for example, the active site which come in contact with the substrate is less than 5% part of the enzyme. So they are very big and provides a large surface for catalytic reactions. All enzymes behaves or work efficiently when their surrounding is favorable. That means they have an optimum condition, and which we generally know as enzyme optima. Enzyme works on a particular pH. They best works on a particular pH as well as on a temperature. So in this way, all enzymes behaves very well with a particular optimal conditions. Enzymes activity can also be inhibited. Okay, enzymes activity can be reduced by some chemicals, and these chemicals are called chemical inhibitors. If we talk about the mechanism, how generally an enzyme works or catalyzes the catalytic activities, the first thing an enzyme performs is that attachment of enzyme with the substrate through its active site. Once the substrate comes in contact with the active site, it forms an enzyme substrate complex which is reversible. And then finally, this enzyme substrate complex undergoes catalytic activity and convert the substrate into product. Once the product is formed, now there is a enzyme product complex, and this product gets detached from the enzyme. So in this way, an enzyme generally catalyzes its activity. There are two methods given by different scientists for enzyme substrate complex formation. The first theory was lock and key theory proposed by Emil Fischer in 1894, and according to this hypothesis, Emil Fischer assumes that the active site of an enzyme are rigid in its shape. That means there is no change in the active site before and after a chemical reaction. There is one specific key for one lock. In the same manner, there is a specific enzyme for a chemical reaction. And the second hypothesis was induced fit model that was given by Daniel Koshland in 1958. And according to him, the exposure of an enzyme to substrate cause a conformational change in enzymatic structure, which causes the active site to change its shape and to allow enzymes and substrate to bind. So that was induced, that an enzyme is getting induced and the site structure also gets induced once it come in contact with the substrate. It changes conformationally itself and the active site becomes fit for the substrate. So there are total two theories given by different scientists. Now we know that an enzyme helps substrate to catalyze with each other to perform chemical or biochemical reaction. But how do they do that? Simply by lowering the activation energy. Activation energy means the least amount of energy needed for a chemical reaction to take place. So whatever amount of energy requires for the substrate to react with each other and to form product, the amount of that energy is now required less once the catalysis is being performed with the help of an enzyme. This is how generally the rate of chemical reaction is increased by using an enzyme, because they lowers the activation energy of that catalytic reaction. Nomenclature, we already discussed that earlier. It was named according to the substrate and by adding the suffix ase. Afterward, the enzyme was named according to the enzyme commission. Each enzyme is described by a sequence of 4 numbers preceded by EC. EC denotes the enzyme commission. When classified, each enzyme is assigned the EC number in the form of digits separated by periods. The first number categorizes the enzyme based on its reaction. Okay, say for example, in the case of EC1, 2, 3, 4, the digits indicate that the enzyme is an oxidoreductase, that is class 1, that it acts on the aldehyde or 2 group of donor, that is class 2, subclass 2, that oxygen is an acceptor, that is subclass 3, and that it was the 4th enzyme classified in this subclass, that is serial number 4. So in this EC 1, 2, 3, 4, EC refers for enzyme commission, 1 shows the class, 2 is for subclass, 3 is for subclass, and 4 is the serial number of that subclass. Classifying enzymes into different categories on the basis of what type of chemical reaction they are participating in. The first is oxidoreductases. They are used to perform oxidation reduction reaction, the transport or transportation of electrons. Transferases especially catalyzes the transfer or exchange of certain groups among some substrates. Hydrolases accelerates hydrolysis of substrates. They works in presence of water. Lysases, they promote the removal of a group from the substrate to leave a double bond reaction or catalyze its reverse reaction. Isomerases generally facilitate the conversion of isomers, geometrical isomers or optical isomers, depends. Ligases generally catalyzes the synthesis of 2 molecular substrates into 1 molecular compound with the energy release. Translocases generally catalyzes the movement of ions or molecules across membranes or their separation within membranes. Let us understand the term enzyme inhibitors and poison that once we already discussed. Those substances which reduces the catalytic activity of enzyme are called enzyme inhibitor. So usually they associate themselves with the active site, and conformational change or some kind of different effect they used to provide on the enzyme so that the enzyme will not be able to catalyze the activities. So it can be done in 2 ways. The chemical inhibitors can do their work in 2 ways. The first way is competitive inhibition, where the structure of inhibitor resembles the substrate structure. In such inhibitors compete with the substrate to combine an active site of enzyme, because of which the enzyme activity decreases. And the another one is non-competitive inhibition. This type of inhibitor permanently attaches with enzyme molecule and bring structural change in it. Now the permanent association denatures the enzyme. Such substances are called enzyme poisons. The catalytic activity of enzyme depends on several factors. Say for example, the substrate concentration. At lower substrate concentration, the active site on most of the enzyme molecules are not filled because there is not much substrate. Higher concentration causes collision between the molecules and the rate of reaction increases. And the vice versa is also applicable. The effect of enzyme concentration is also seen, like as the amount of enzyme is increases, the rate of reaction increases. If there are more enzyme molecules than are needed, adding additional enzymes will not increase the rate. Reaction rate therefore increases as enzyme concentration increases, but then it levels off. Effect of product concentration, that the enzyme activity deadlines or declines when the products start accumulating. This is called product or feedback inhibition. Effect of temperature is also seen, as we know that enzymes are proteinaceous in nature and protein can be denatured above an optimum temperature. So the effect of temperature is obviously very logical. The effect of pH is also seen. All enzymes works efficiently at a proper pH, whatever they needs. The effect of inhibitor we already discussed, and here we are done with this session. I hope you will understand all the concepts related to this enzyme chapter. I'll be here soon. Have a nice day.